Welcome. My name is Réka Hollandi. I'm from Péter Horváth's lab Biomag at the Biological Research Center in Szeged, Hungary, and I'm going to talk about nucleAIzer, a method for nucleus segmentation with deep learning and image style transfer. I don't suppose I need to introduce deep learning to you, so let's begin with the task. On this example image, we want to identify the purple objects, the nuclei, on the bright background here. We can either only detect them, defined by these bounding boxes, which is enough for simply counting. But if we need more information, then we need to segment the objects themselves, that is, identify the area corresponding to each individual object, and also separating the objects from each other so that we can conduct specific measurements like morphologies, texture, and intensities based on the objects themselves. Or we can do both. Let's see what are the main issues in this task. Firstly, experimental conditions tend to rapidly change from one experiment to the other. If we consider these 5 examples here, we can see that these two correspond to histological tissue sections imaged in bright field mode, and the others are fluorescent cell culture images. And even if we repeat the same experiment with the very same conditions, the imaging part or sample preparation might cause differences, or simply the cells themselves sometimes tend to rapidly overgrow compared to other experiments. So we need software solutions that can cope with these differences. Next, in typical high throughput experiments, the amount of data we need to process can contain many plates, even thousands of images, and even terabytes of data, so we need software solutions that can cope with that. Also, very typically in our biological image processing area, we need to deal with cancer cells, which tend to grow on top of each other. If you consider the bottommost example here, you can see from the green accumulation area here that the cells heavily overgrow each other, which causes occlusions and makes it really difficult for an image processing task or solution to tell the individual objects apart from each other. And last but not least, we need expert knowledge to be able to accurately define what is an object and what is not. For example, on this tissue image here, we know that these are red blood cells actually, and not nuclei, so we shouldn't identify them as such. But for an algorithm, they look kind of similar because they are elliptical shape and possess some color. Fluorescent images might be stained red, so it might be difficult for them to identify them as non-nuclei objects, and also in the middle example, these smaller objects here might be actually artifacts that also shouldn't be identified as nuclei, but we need the experts to teach the model, teach us what is an object and what is not, and this expert knowledge is expensive and time-consuming. So, to sum it up, we need methods that are robust, reliable, and accurate, and most importantly, work well on our own experiments, because in the end, that's all we care about. Let's see what our idea was. Suppose that we have a new experiment that we've never worked with such samples before, and the images look like this. We can see that this is a tissue with a rather complicated background. Since it's a tissue, nuclei tend to fade into the background because they are not cut at the right Z position in the volume. So it is rather difficult for an image processing algorithm to accurately identify the nuclei. So what we thought to do was to generate a synthetic binary mask where the white areas mark the nuclei and everything else is the background, and we want to simulate nuclei in these white areas. So we did that so that we can have new synthetic microscopy images with the faithful texture representation of the original style, both for the foreground and the background. The background is also important for training purposes. And you can see that these 3 objects correspond to these masks here. With this method, we generate as much of these images as we want to use them for training, and with this method, compared to conventional segmentation algorithms, we can perform much better. You can see that the conventional algorithm fails to identify these regions on the image as background, and also the separating lines and the true boundaries of the objects is difficult for the method. But for our method, it looks much better. We can see that most of the objects are nicely detected, and also the contours look accurate. So with this synthetic image generation approach, which is called image style transfer, we can satisfy two important aspects needed to be considered here. One of them is that a large training set is needed for deep learning methods to perform accurately, and since we can generate as many of these images as we want, because we also generate the masks which form the basis of the synthetic images, we can meet this expectation and also include new experimental condition information in the training, which is highly desirable. We dubbed this method nucleAIzer, and it is an instant segmentation pipeline using image style transfer. We also created an online tool for it that you can find at nucleAIzer.org. And how we use this for image segmentation is shown in this figure. We consider our previous experiments as prior information with corresponding ground truth annotations, and when new experiments come in that we have no idea how to process and don't know how to identify the nuclei, we adapt to their style, create new synthetic images in their style as I showed you, and use these images to train segmentation, also using the previous experimental information, so that we can have one segmentation model that can accurately and robustly identify the nuclei on all of these images. So if you like, it's a one-fits-all solution. Let's see how this pipeline works. Panel A shows the entire pipeline, and B the style transfer part. Let's begin with the pipeline. The input images are the heterogeneous microscopy images from various experiments. First, we do a pre-segmentation step using mask-RCNN, which is an instant segmentation network. And we use these masks for size information retrieval in subsequent steps, such as for augmentation. Here comes the style transfer part, for which we use pix-to-pix implementation. We begin with a heterogeneous set of original images that can contain cell culture images, tissue images, acquired in bright field of fluorescent mode, different stainings, different cell lines, etc. And based on their visual appearances, we cluster them to different groups. And for each of these clusters, we build a style model. We begin with the original images and their corresponding binary masks that might come from previous annotations or databases, or from the pre-segmentation step. And we train a so-called style model that learns how to create a realistic-looking intensity image like this from the binary mask. And simultaneously to this, we also generate synthetic masks, for which we use our previously assembled nucleus mask database, and select such individual nucleus masks that resemble the morphology of the original images, and place them in such a fashion that the spatial distribution and size of the objects follow that of the original images, and create such synthetic masks. So for each cluster, we have a trained style model, and we have new synthetic images. So now we only need to apply the model on these masks to generate the artificial microscopy images, as you can see here. Since we can generate as much as we want, we can effectively boost our training set, and it can be also useful to emphasize rare types of images that were not properly represented in the training set before. Let's play a game. Can you tell me which one of these images is potentially one that we generated with the synthetic image generation method that I showed you? Well, the answer is they're all fake. We created these masks, as I just described, trained a separate model for each of them, and then applied them on these on these masks so that we can have these nice synthetic images, and we use them for training. So that's where we were. Of course, we also use conventional augmentation techniques like flipping, rotation, resizing, cropping, and so on to further boost the training set. Then train a Muscar CNN network for segmentation, and then we can use it to actually segment the input images, for which we use the size information retrieved from the pre-segmentation step. And here, as the gray dotted line suggests, we can iteratively retrain our model using the newly trained model as a pre-segmentation model, so that we can have more accurate initial masks and better looking style transfer images and so on. If we are satisfied with the segmentation results here, we can fine-tune them with post-processing, where we use UNet and classical morphology operations to generate the output masks themselves. For the post-processing, on panel A you can see a scheme, and on B a real example. I'm going to show the steps on A. This red contour represents the Muscar CNN result. This is extended and shrunk by a few pixels to generate a soft margin around the objects. Then UNet models are trained, and their prediction is used within this soft margin to create a final corrected segmentation for each object. You can see with the green line here, and on C a real example for a rather difficult tissue image. You can see that it performs quite well. Then let's see a few results. We originally created this method for the data science born nucleus detection competition from 2018, where the test set contained some truly challenging images, as you can see presented in a few examples here. And the task was to create one method that can cope with all of them in once. And you can see the results of our method overlaid in red on these images. I believe we did quite a decent job there, and our method is currently the best performing one. On this dataset, the competition consisted of two test sets. You can see a few examples here, where we wanted to show that different sizes, shapes, and intensities are no match for the method. A few examples: a typical artifact in fluorescent images is this staining artifact that doesn't confuse the model, and also these crowded areas on the tissue can be solved nicely, and also the dividing lines are faithful. And of course, we also applied this method in our own experiments in the lab and our collaborators. The first is from our lab, a mouse kidney tissue immunohistochemically stained with the brown marker for cancer cells. But for proper quantification, we also need to identify the healthy cells, which are marked in blue. You can see that they fade into the background quite strongly, so it is rather difficult to see them even by eye. But if I overlay our result, then you can see that it's quite nice. The next project is a collaboration with Hella Bork at the University Hospital Zurich. This is a kidney cancer tissue from a patient, hematoxylinosin stained with rather complex structures and crowded regions like here. You can see that we can solve this task as well quite nicely. The next project is with the Matthias Mann group at the University of Copenhagen and Max Planck Institutes in Denmark and Germany respectively. This is an example UTO's cell culture fluorescently stained for actin and tubulin, and this time we wanted to also identify the cytoplasm, so we trained a different model for that. I also displayed the nucleus detections here, but it was rather easy on this nice image. The next sample from the same group is an ovarian cancer tissue, this time fluorescently labeled for the cell membranes in red, as you can see here, which is a bit more difficult this time. Our results are overlaid in white. You can see that even in these faintly visible areas, it does quite a nice job. The next project is a collaboration in our host institute with Victor Honti. These are insect cells fluorescently labeled for differentiation markers in green and red, overlaid on the bright field channel, and we wanted to identify the cytoplasms here. In the next project, which is a collaboration with the Leukemia Research Group at University Children's Hospital Zurich. We had leukemia and bone marrow cultures fluorescently stained, and we wanted to identify these small bright cells, which are the leukemia cells, together with the healthy stroma cells, the dimmer bigger ones, for subsequent classification then, and did quite well there as well. The next project is with the Kutai group at the ETH Zurich, where we did a genome-wide screen for different RNA markers, and the example image shows here nucleoli staining, which are these small brighter dots inside the nucleus. So we also trained a new model for these ones, which you can see overlaid in red here, and the nuclei in green. You can see that we could also quite nicely identify these ones. And finally, another collaboration with the Yohei Yamauchi at the University of Bristol in a coronavirus project, where we wanted to identify the nuclei in the syncytium areas, which are these large fused cells here. And we did quite well there as well. For the paper, naturally, we performed proof of concept experiments on both histology and fluorescent image sets, and found that our method performs better than the others. But let's see a more interesting example here. We also compared our results using and not using the stack transfer method to show that it actually does help on these rather difficult example images here. So let's see them one by one. The upper one will be without style, and the lower one with style. I'll highlight the differences. Here we got rid of this false detection area here. You can see in blue. Here we found an extra object that originally was missed, and here, unfortunately, we could detect only a smaller portion of the object that was better without style for some reason. The next image shows that these two cells were missed without style, and after style information in the training set, we could detect also most parts of this object. And also here we detected two objects. Here you can see the dividing line, while here it was only a portion of it fused. And also we had another extra that was originally missed. This crowded fluorescent and out of focus image, we could also detect an extra object, but unfortunately introduced false detection. And in the final one, we could better identify this dividing line between the objects when using style. So, as a summary, in most cases, it actually did help. As well as this all may sound, you might still wonder why you would want to use this method over the many others that exist. Well, let me refer back to my initial slides about the issues. First of all, expert knowledge is needed to use these widely used bioimage analysis software like Fiji Elastic or Cell Profiler. You need to know which modules to or which modules to use in your project, and how to set the parameters. And also, since experimental conditions can change significantly, with each of these changes, you need to refine your parameters and modify the pipelines, which is undesirable. So for a typical end user who doesn't want to spend much time on that part, they just want to forward their images to something and get the outputs on the other end. You can think of this method as a black box, and forward a folder of input images and get the output on the other end, just like that. Of course, more experienced users or developers can look into it, see how it works, which is the pipeline I just described, modify it to better fit their experiments and their needs, see what kind of technology we use there, and most importantly, check out the GitHub page of the project and share your comments or remarks with us, also issues if you have them. They're all very welcome. And last but not least, how do you really get started with this? Because eventually, what's most important for all of us is that any method should work well on our own experiments. For that purpose, we created this online tool you can access at nucleAIzer.org, where you can drag and drop to upload your own images, select a model that best represents your own experiments, or else just using the general one, which is supposed to work on any new experiment, and get the results right away, so that you will actually know if it's worth your time or not. And as a final thought, I would like to take this opportunity to advertise my other project, which you can think of as an alternative to the style transfer approach of introducing new experimental information in your project via annotation, of course. We created a Fiji plugin called Annotator.j that uses a built-in pre-trained deep learning model for contour suggestion to efficiently and fastly annotate new images and generate masks that can be used to train different kinds of deep learning models, and so it also nicely can be prepended to the nucleAIzer method that I just described to generate the masks. Last but not least, I would like to thank all the people who contributed to this project, mainly Péter Horváth's groups in Hungary, Biomag, and in Finland, FIM, mostly Abel, Ervin, Csaba, Tivadar, and András in Hungary, and Lassi in Finland, and also our external collaborators, Kevin from Sweden, and Anne Allen and Juan from the Broad Institute. And with that, I thank you for your attention.